Hi everyone. So today we are going to look at the differences between two types of microscope, the light microscope and the electron microscope. But before we do that, a bit of retrieval practice. Pause the video here and have a go at these 10 questions. Great. So hopefully you've had a go at all of those, and let's see if you've got those answers right. So question 1. 5 subcellular structures that are found in both plants and animals. Cell membrane, cytoplasm, nucleus, ribosomes, and mitochondria. Fantastic, well done. 3 subcellular structures found in plants but not in animals. Cell wall, vacuole, and chloroplast. Make sure you've got those marked. Question 3. Give 2 differences between prokaryotic and eukaryotic cells. So there's no nucleus in prokaryotic cells, and they also don't have membrane-bound organelles. Question 4. The function of the cell membrane is to allow substances to pass in and out. The function of cytoplasm is where the chemical reactions take place. Question 6. Green organelles found in plant cells are called chloroplasts. The function of these organelles is basically where photosynthesis takes place. They also contain chlorophyll, which is the pigment that makes them green. Root hair cells are adapted to their function because they have a large surface area. This maximises the absorption of water. They also have tiny hairs on the surface, which again creates an even larger surface area to absorb all that water. Question 9. Red blood cells are adapted to their function because they have no nucleus. They are circular in shape, but they have like dints in the middle. We say this is a biconcave shape. This maximises the oxygen carrying capacity of the red blood cell. They're also small enough to pass through capillaries at about 7 nanometers in diameter. And sperm cells are adapted to their function because they contain an acrosome. This is found in the head of the sperm cell, and it helps to get into the egg cell because it is able to digest the outside of the egg. They also have lots of mitochondria to provide them with lots of energy to swim, and the tail helps them move in a particular direction. So, how did you do? Hopefully you got all of those right. Well done. So our success criteria for today then. We want to identify the features of both our light microscope and our electron microscope. We want to describe these features and compare the two microscopes. And finally, evaluate images to explain which type of microscope can produce the image. Our keywords then are micrometer, which is a unit of measurement even smaller than a millimeter. The resolution is how clearly you can distinguish between 2 points. Magnification, being able to make an object appear larger. And by the end of this session, you'll be able to give me a definition of what an electron microscope is. So, your first task then. Pause the video now and have a go at labelling the microscope. So, on your sheet of paper that you've got in front of you, number your page 1 to 14 and have a go at naming the different parts. Pause the video now to help you do this. Done? Great. So, question 1, or part 1 as I should say, is the body tube. Number 2, the resolving nose piece. Number 3 is the low power objective lens, and as you can see there's 2 more. Number 4 is medium power objective lens, and 5 high power objective lens. 6, we've got our stage clips. 7, we've got the diaphragm. Diaphragm just allows light to enter or leave the specimen, and we can adjust how much light enters or leaves using the diaphragm. 8 is our light source. 9 is our eyepiece. 10 is the arm. 11 is the stage where the specimen goes. 12 is the coarse focus. We adjust that first before adjusting 13, which is our fine focus. And 14 is the base. Well done. So, 2 different types of microscope. Hopefully you've all seen a light microscope in school and had a look at some cells. We don't have an electron microscope in school. Can anyone guess why? You got it. It's too big and too expensive. At one time, these electron microscopes would take up their whole room. They're usually found in universities or hospitals. So the next 2 slides on this clip contain information on the electron and the light microscope. While pausing the video, I'd like you to find out about these 7 points. So, what each type of microscope uses to see the cells, how the specimens need to be prepared so we can view them, what the resolution is for each microscope, what the magnification is for each microscope, and advantages and disadvantages of each. Also, find out what each type of microscope can see. So you can rewind the video at any point to go back to these questions. So, compound light microscopes, the ones we have in school, are the oldest and simplest and most widely used form of microscopy. Specimens are viewed using light, which is focused using the eyepiece lens and the objective lens. Slides are viewed using the eye or as a photo connected to a computer. The specimens under a light microscope can be living or dead, but they often need to be stained with a coloured dye to make them visible. All of our light microscopes today are called compound microscopes, which means they use several different lenses to obtain a higher magnification. The highest magnification would be about 1500 times. However, this is not as good as an electron microscope. We'll look at that in a bit. Light microscopes have a resolution of about 200 nanometers, which is good enough to see cells, but not the details inside cells such as organelles like mitochondria, ribosomes and chloroplasts. Light microscopes are popular as they're cheap, they're easily transported, and they're not too heavy either. Moving on then, let's have a look at electron microscopes. Electron microscopes use a beam of electrons rather than a wave of light to view the image. The electrons behave like a wave and the images are viewed on a screen over here on a computer. So this is our electron microscope. These are eyepieces if a person would like to view it in that way, or we can see the results on the computer screen here. They have a really high magnification and a very high resolution, about 1 nanometer, allowing organelles such as mitochondria, ribosomes and chloroplasts to be seen in detail for the first time. Some electron microscopes are able to produce 3D images, which are not possible with a light microscope. The main problems with electron microscopes are that the specimens must be fixed in plastic and viewed in a vacuum with no air, and therefore they must be dead before we can view them. Other problems are that specimens can be damaged by the electron beam, and they must be stained with a heavy metal such as gold or lead to protect them. This can sometimes produce artifacts, which are small pieces of metal that are not part of the cell and give a false image. Other problems with the image is that it's in black and white, but like the picture at the bottom of the egg and sperm here, we can give them false colours so that we can make them appear more interesting and easier to see. So if you've unpaused the video, you can always go back and check that you've got all the information you need from these 2 slides. Once you've done that, we can move on. Brilliant. So, using those notes that you've hopefully made, I'd like you to create a table with an electron microscope on one side, light microscope on the other, and then write the different boxes on here underneath the correct headings. If you find that really easy, then there's a challenge question in the top corner there for you to have a go at. Pause the video now and do this for me, please. Done that? Fab. Let's see if you've got those in the right places then. So the electron microscope has the higher magnification and the higher resolution. It's more expensive, it uses electrons to form the image, and the specimens must be carefully prepared and will not be living. The light microscope's got a lower magnification and resolution, it's cheaper, it uses light and can view living specimens. Got that sorted? Fab. So, in this session, we've been able to identify the features of microscopes, describe those features and compare them. So, lastly, can you evaluate the images to explain what type of microscope can produce which image? Have a go and see if you can. So, which structures can the light microscope resolve so we can see them? And which can the electron microscope resolve? So we said that the resolution of a light microscope would allow us to see cells and possibly some organelles, but not very clearly, while our electron microscope would allow us to see bacteria, viruses, cell membranes, and even down to the atomic level, sometimes even atoms. So have a go with these questions for me then, please. Pause the video now. Okay, so can we find mitochondria in bacteria? Justify your answer using evidence. So look at the size of a mitochondria. It's approximately 10 micrometers, but a bacteria is only 1 micrometer, so a bacteria is far too small to have mitochondria inside it. So here we've got 2 images. Which do you think is from a light microscope and which is from an electron? Okay, so the pink one here is muscle cells, and this is stains, so we know straight away it's from a light microscope, and this is black and white, so it's from an electron. We can also see this is like a slice taken through the cells, and so this clearly gives us an idea that it's from an electron microscope. So that's the end of today's session. And hopefully you now can know the difference between the light and the electron microscope. What I'd like you to do now is write down a definition of what an electron microscope is. Great, well done.